we come to biology, the scientific study of life. More about enzymes. Effects of local conditions on enzyme activity. The activity of an enzyme, how efficiently the enzyme functions, is affected by general environment factors such as temperature and pH. It can also be affected by chemicals that specifically influence that enzyme. Effects of temperature and pH. Recall from chapter 5 that the three-dimensional structure of proteins are sensitive to their environment. As a consequence, each enzyme works better under some conditions than under others, because these optimal conditions favor the most active conformation for the enzyme molecule. Temperature and pH are environmental factors important in the activity of an enzyme. Up to a point, the rate of an enzymatic reaction increases with increasing temperature, partly because substrates collide with active sites more frequently when the molecules move rapidly above the temperature. However, the speed of the enzymatic reaction drops sharply. The thermal agitation of the enzyme molecule disrupts the hydrogen bonds, ionic bonds, and other weak interactions that stabilize the active conformation, and the protein molecule eventually denatures. Each enzyme has an optimal temperature at which its reaction rate is greatest without denaturing molecular collisions and the fastest conversion of reactant to products molecules. Most human enzymes have optimal temperature of about 35 to 40 Celsius degrees, close to human body temperature. Bacteria that live in hot springs contain enzymes with optimal temperature of 70 Celsius degree or higher than. Just as each enzyme has an optimal temperature, it also has a pH at which it is most active. The optimal pH values that for most enzymes fall in the range of pH 6 to 8, but there are exceptions. For example, pepsin, a digestive enzyme in the stomach, works best at pH 2. Such an acidic environment denatures. Notice this picture or diagram. Most enzymes but the active conformation of pepsin is adapted to maintain its function, a three-dimensional structure in the acidic environment of the stomach. In contrast, trypsin in a digestive enzyme residing in the alkaline environment of the intestine has an optimal pH of 8, and notice that and would be denatured in the stomach. Notice this also optimal temperature of two enzyme temperature for notice the temperature of human enzyme or typical human enzyme for between 20 and 40 25 Notice also temperature optimal temperature enzyme thermophilic bacteria, notice 80 and more. Notice also pH, notice also the pH optimal pH 2 for human or the pepsin stomach enzyme. For optimal pH for trypsin intestinal enzyme is 8. Okay. Cofactors. Many enzymes require non-protein helpers for catalytic activity. These adjacent, called cofactors, may be bound tightly to the enzyme as permanent residents. Or they may bind loosely and reversibly along with the substrate. The cofactors of some enzymes are inorganic, such as the metal atoms zinc, iron, and copper in ionic form. If the cofactor is an organic molecule, it is more specifically called a coenzyme. Most vitamins are coenzymes or raw materials from which coenzymes are made. Cofactors function in various ways, but in all cases where they are used, they perform a crucial function. In catalysis, we will encounter examples of cofactors later in the book. Enzyme inhibitors. Certain chemicals selectively inhibit the action of specific enzymes, and we have learned a lot about enzyme function by studying the effects of these molecules. If the inhibitor attaches to the enzyme by covalent bond, inhibition is usually irreversible. Many enzyme inhibitors, however, bind to the enzyme by weak bonds, in which case inhibition is reversible. Some irreversible inhibitors resemble the normal substrate molecule and compete for admission into the active site. Notice this. Called competitive inhibitors reduce the productivity of enzymes by blocking substrate from entering active sites. Notice that. This kind of inhibition can be overcome by increasing the concentration of substrate so that as active sites become available, more substrate molecules than inhibitor molecules are around to again entry to the sites. In the contrast, non-competitive inhibitors do not directly compete with the substrate to bind the enzyme at the active site. Instead, they impede enzymatic reaction by binding to another part of the enzyme. This interaction causes the enzyme molecule to change its shape. Notice that. This interaction causes the enzyme molecule to change its shape, rendering the active site less effective at catalyzing the conversion of substrate to product. Toxins and poisons are often irreversible enzyme inhibitors. An example is sarin, a nerve gas that caused the death of several people and injury to many others when it was released by terrorists in the Tokyo subway in 1995. This small molecule binds covalently to the R group on the amino acid serine, which is found in the active site of acetylcholinesterase, an enzyme important in the nervous system. Other examples include the basticides, DDT, and the parathion, inhibitors of key enzymes in the nervous system. Finally, many inhibitics are inhibitors of specific enzymes in bacteria. For instance, penicillin blocks the active sites of an enzyme that many bacteria use to make their cell walls. Citing enzyme inhibitors that are metabolic poisons may give the impression that enzyme inhibition is generally abnormal and harmful. In fact, molecules naturally present in the cell often regulate enzyme activity by acting as inhibitors. Such regulation, selective inhibition, is essential to the control of cellular metabolism, as we discuss next. Okay, thanks for listening.